Hello, my name is Dorothy Nediak. I am 15 years old and I study at the International Gymnasium of the Skolkovo Innovation Center in Moscow, Russia. Today, I will show you how yeast and bacteria in symbiosis create fermented kombucha tea. What is kombucha tea? Well, we start with sweetened tea, inoculated with a SCOBY, which stands for Symbiotic Culture of Bacteria and Yeast, and together with accompanying starter liquid, which can be active kombucha or just vinegar if active kombucha is unavailable, to avoid mold by bringing the pH down. And then this is fermented in a warm, dark place, first aerobically, and then if higher CO2 concentration is needed, anaerobically in sealed bottles. And how does it actually work? Inside the SCOBY is a cellulose matrix that is made by the bacteria. The bacteria take glucose and make cellulose. The exact species of bacteria and yeast in the SCOBY vary depending on the exact SCOBY and the location, which is kind of cool because unlike other types of fermentation where there's a very specific species, in kombucha it really varies. These are some of the more common species. What the microorganisms actually do. Yeast takes glucose and makes CO2 and alcohol, but kombucha is very low in alcohol, and that's because the bacteria take the alcohol, oxidize it, and get acetic acid. And this is where we go from oversweetened tea to sour, fizzy kombucha. Now, while these chemical changes are occurring, the microbial colony also visibly changes. In the middle of the brew, new yeast strands grow throughout the fermentation. Looking at these new yeast strands, we see translucent filaments, and we can also see some sort of cells around the microfilaments. Also, at the bottom of the jar, dust of some sort accumulates. It's very fine, it scatters very easily, and for that reason it is difficult to measure, but it is still clear that it increases in volume over the course of the fermentation. Looking at this dust under the microscope, you can see that the particles are very thin and fine, flaky particles. The structure of the SCOBY. The main bulk of the SCOBY is uniformly colored, beige-ish cellulose, but on the surface there is a clearer layer and yeast is also attached to it here, as this picture very clearly shows. These pictures have yeast over the main cellulose layer. The surface of the brew. Usually, a baby SCOBY will grow on the surface of the brew, a new pellicose disc. However, that did not happen in my case, which could be due to low temperature or inadequate oxygen. However, after 6 days of storage in active kombucha, the mother SCOBY in the jar began to grow a baby SCOBY. So the culture itself is active, and the results were sour enough, demonstrating bacteria activeness, and fizzy enough that there was actually not a need for secondary fermentation, but it was ready after 7 days of primary fermentation, and it was good kombucha. Thank you to Universidad del Norte for hosting this event, giving me this opportunity. Thank you to my supervising teacher, Tamara Banyevich, for supporting me with this project. And thank you for watching.